Topic 4 Exchange and Transport. 4.1 Cell Transport Mechanisms. Explain how passive transport. Active transport requires energy, so passive transport involves no energy or ATP. Passive transport is brought about by simple diffusion, facilitated diffusion, and osmosis. In simple diffusion, particles move from a higher concentration to a lower concentration down a concentration gradient. Facilitated diffusion is the same as simple diffusion, except that diffusion takes place through a transporter protein, which could be either a channel protein or carrier protein on a membrane. Again, the particles move down a concentration gradient across the membrane. Proteins have got 3D shape because they are globular. They also have a charge due to their R groups being polar. Therefore, the shape or charge of a protein can affect facilitated diffusion, as it only allows specific substance to move through. For instance, if you have a potassium ion channel protein, only potassium ions can diffuse through the channel protein. So different channel protein and carrier protein will diffuse different particles based on the shape and charge of the transporter protein. Osmosis is the movement of water down a water concentration gradient, for instance from a higher water concentration to a lower water concentration, or it's the movement of water from a lower solute concentration to a higher solute concentration through a partially permeable membrane. And here you can see an example of osmosis, where you've got higher water concentration on the left side, and the water is moving to a lower water concentration on the right through the partially permeable membrane. On the left, you could see that there is no sugar or solutes, so there's a lower solute concentration. So using the same idea, you could also say osmosis is the movement of water from a lower solute concentration to a higher solute concentration for a partially permeable membrane. And here's an example of passive transport. We've got osmosis, diffusion, and facilitated diffusion through transport protein, channel or carrier on the membrane, as opposed to active transport or endocytosis, which both requires energy in the form of ATP. State structure of cell surface membrane with reference to fluid mosaic model. The membrane consists of phospholipid bilayer that also contains channel and carrier proteins. In the phospholipid bilayer, not only do you have phospholipids, but you've got peripheral proteins which are on the surface of the membrane. There are channel and carrier proteins, and they are integral proteins because they cross the whole membrane, so they're transmembrane. You've also got cholesterol, which reduces the fluidity of membrane, so it makes it harder for the phospholipids to move around in locations where there are cholesterol. And you've also got glycoproteins, which are carbohydrates attached to a protein. The phospholipids themselves form a bilayer, and on the inside you can see that there are fatty acid tails, and that's because these fatty acid tails are nonpolar, so they are hydrophobic. They can't form hydrogen bonds with water, so therefore they like to be on the inside. On the outside of the phospholipid bilayer you have got phosphate heads, and that's because these phosphate heads are polar and can form hydrogen bonds with water, so they are hydrophilic. Explain how properties of molecules affect how they are transported, including their size, charge, and solubility. Small or large nonpolar or lipid-soluble molecules, such as oxygen or carbon dioxide, are hydrophobic. So therefore, they like to enter via the phospholipid bilayer membrane by simple diffusion. Those molecules that are small or charged, such as polar or ionic, or water-soluble molecules, they are hydrophilic, so they don't pass through the hydrophobic fatty acid tail center. Instead, they have to be transported using either channel proteins, which happen through facility diffusion. An example of this is water by osmosis, or they could use carrier proteins, which also uses facility diffusion, but can also be involved in active transport. Extremely large molecules that are too big to fit through the channel or carrier protein, including proteins and even viral genetic materials, are transported in vesicles by endocytosis or exocytosis. Core Practical 5. Investigate effect of temperature on beetroot membrane permeability. Beetroots contain a pigment inside their cells, so if you disrupt the membrane, that pigment can then diffuse out. The method involves cutting equally sized pieces of beetroot. That is a control variable. You should also rinse it underwater, and that's to make sure that there is no beetroot pigment that is released before you start the experiment, which affects the accuracy of the results. You'd place them in a range of different temperatures, which is your independent variable that you're changing, which you need at least 5 different types. You'd leave them for equal time, and that is a control variable. And then you would assess how much pigment is lost using a calorimeter, which measures the absorbance or the transmission. When the temperature affects the membrane permeability, you can then plot a graph on the temperature on the x-axis and absorbance or transmission on the y-axis. And you should get a line of best fit or curve like this, where increasing the temperature increases membrane permeability. And this is because the phospholipids have more kinetic energy when they've got a higher temperature. This allows them to separate from each other more, that leaves more gaps for the pigment to be released. And the more pigment that is released, the more darker the color that is observed in the colorimeter. Explain effect of increasing ethanol concentration on membrane permeability. Ethanol has a similar property where it also damages phospholipids, and that increases the permeability and allows pigments to go out. Another way in which temperature could affect membrane permeability is that not only does it separate the phospholipids, but it could also denature the membrane proteins, and again that increases permeability and allows pigments to go out. Explain water potential of plant tissue. In hypertonic solutions, this is where there's a higher solute concentration in the solution. When you place an animal cell in a hypertonic solution, it starts to shrivel because water moves out of the animal cell and into the hypertonic solution where there is a lower water concentration. In a plant cell, the same thing happens. Water moves out of cells by osmosis, but the cell becomes plasmolysed. And this is where the cell membrane detaches from the cell wall. In isotonic solution, there is the same solute concentration in the solution and inside the cells. Therefore, there is no net water movement. The amount of water moving out of a cell and into a cell by osmosis is the same, so the overall net movement is 0. In a hypertonic solution, there is a lower solute concentration in the solution. Therefore, there must be a higher water concentration in the solution and a lower water concentration in the cell, so water moves into the cells by osmosis, and this will cause an animal cell to lyse. However, a plant cell will become turgid because it has a cell wall that prevents lysis from happening. A cellulose cell wall prevents plant cells from lysing or bursting. Plasmolysis is when the cell membrane pulls away or detaches from the cell wall. We can measure this by calculating the incipient plasmolysis, and that is the solute concentration when 50% of cells are plasmolysed. Water potential equals turgor pressure plus osmotic potential. Water potential is the potential for water to move out of solution by osmosis. So pure water has the highest water potential of 0 because it's got the highest potential to move out from a solution. Every other water potential for solutions has a negative value. Turgor pressure is the inward opposing pressure exerted by the cell wall against any expanding cell contents, and this is usually a positive value. So as a cell becomes more turgid, the turgor pressure increases, and this reduces the ability of water to move into a cell by osmosis. The osmotic or solute potential is the potential of water to move across partially permeable membrane from a hypertonic solution to a hypertonic solution. So when there's a greater solute concentration in a solution, they have a lower osmotic potential, and the value is always negative. So any cell that has a high osmotic potential, it means that most likely water will move out of it to a lower osmotic potential solution. The water potential of animal cell is osmotic potential of the cytoplasm. That is because animal cells do not have any cell wall, so there is no cell wall exerting hydrostatic pressure. Therefore, turgor pressure has to be 0, so the water potential equals the osmotic potential. Using this, we could have a new osmosis definition for A level, which is the movement of water from a higher water potential to a lower water potential, or down a water potential gradient across a partially permeable membrane. You should use this definition for exams. Here's an example where in distilled water you've got a water potential that is equal to 0. When you put a plant cell immediately into distilled water, there's a higher water potential outside, so water moves into the plant cell by osmosis. As it keeps moving in, the cell contents push against the cell wall, and that increases the turgor pressure until the turgor pressure is the same as the osmotic potential and no more water can move in by osmosis. Explain process of active transport. Active transport is transport particles from a low to high concentration against the concentration gradient. This requires energy in the form of ATP, and it also involves carrier proteins, which change shape using energy from the hydrolysis of ATP. An example of this is mineral ions that are actively transported from the soil to plant root hair cells. Describe how ATP provides energy. ATP is short for adenosine triphosphate, and it contains an adenine nitrogen space, a ribose sugar, and 3 phosphates. The energy in these phosphate bonds makes ATP a universal energy supplier because it provides accessible supply of energy for biological processes such as DNA replication, protein synthesis, and also metabolic reactions that occur in the cytoplasm. When you break the third phosphate bond, that releases huge amounts of energy. So hydrolysis of ATP involves breaking that third bond, and that provides accessible supply of energy. ATP is made by phosphorylating ADP, but this requires an inorganic phosphate and lots of energy, which is found through respiration. The energy released by respiration is used to phosphorylate ADP to make ATP, and then that ATP is used as an accessible supply of energy for other reactions. Describe how large molecules can be transported into and out of cells. This involves either endocytosis, such as phagocytosis, or exocytosis. In endocytosis, the membrane engulfs outside molecules because it is fluid. It then pulls inwards and pinches off to form a vesicle. And this is an active process as it requires energy from the hydrolysis of ATP. The vesicle then moves in towards the cytoplasm. In exocytosis, the vesicle fuses with the membrane, which also requires energy from the hydrolysis of ATP, and this releases the content of the vesicle, such as proteins or other large macromolecules, out of the cell. And here's an example of both active transport and endocytosis, which is an active process that requires the hydrolysis of ATP to release energy. Note that in active transport you can see that the particles are moving against the concentration gradient. Whereas in the endocytosis animation, you could see the fluidity of the membrane when it engulfs the surrounding particle and forms the vesicle. What's the solute concentration of potato cells tissues? This is a practical that was done in GCSE biology and is still relevant for A-level. First, you have at least 5 different potato chips. You find out their mass and record them. That is the initial mass. You then put them into at least 5 different sucrose solution concentrations. In some of these potato chips, water moves in towards the potato chip from the solution and increases their mass. Therefore, there must have been a high water concentration in the solution and a lower water concentration in the potato chips. Since a high water concentration is the same as a lower solute concentration, that means that the sucrose solution must have had a lower solute concentration and the potato cells must have had a higher solute concentration. So using the same idea, water must have moved in from a lower solute concentration in the sucrose solution to a higher solute concentration in the potato chips. In other potato chips, the water will move out and their mass will decrease. This is because there's a high water concentration in the potato cells and a lower water concentration in the sucrose solution. Therefore, water will move out by osmosis. And using the same idea, we could also say that the potato cells therefore must have a lower solute concentration, and in those solutions they have a higher solute concentration. So water moved out by osmosis from a lower solute concentration in the potato to a higher solute concentration in the sucrose solution for those experiments. We could then figure out what the solute concentration of potato cells is, because we could see that it's between 0.4 molar where there was a mass gain and 0.6 molar where there is a mass loss. That's because that's where we expect there to be no change in mass. We then have to record the final mass of the potato chips, and that involves blotting the potato to remove any excess fluid. And using the initial mass and the final mass, we could then calculate the percentage change in mass, which is final mass minus initial mass divided by initial mass and times 100, as is a percentage. Note that some of the values will be negative, and that's because there's been a loss in mass, whereas a positive value signifies that there is a gain in mass. We can then plot all these results onto a graph. So on the x-axis, you've got the solute concentration. On the y-axis, there's the percentage change in mass. You can see the trend is as you increase the solution concentration, the percentage change in mass decreases. From 0 to 0.46 moles, we could see that there is a percentage gain in mass, and that's because water moved into the potato chips from a higher water concentration in the solution to a lower water concentration in the potato. Above 0.46 moles, we can see that there is a percentage loss in mass, and that's because water moved out of the potato and into the solution from a higher water concentration in the potato cells to a lower water concentration in the sucrose solution. And we can see at 0.46 moles, there is a 0% change in mass. Therefore, there must be no overall gain or loss in mass at that concentration. And this tells us that the solute concentration of the potato must therefore be 0.46 moles. Core Practical 6, determine water potential of plant cells. So to devise an experiment to compare water potential of carrot and potato tissue, clearly we used a range of at least 5 different sucrose or salt concentrations. We had equal surface area of the carrot and potato pieces. That is a control variable. We also have another control variable where we have to control temperature, as it affects the movement of water by osmosis. You should submerge the carrot and potato pieces in the different solutions so that every part of the surface is in contact with the solution for the same amount of time. This is another control variable. The carrot and potato pieces have to be weighed before and after submerging in solution to find out their initial and final mass. And when working out the final mass, you should blot the pieces dry before weighing them to remove any excess solution on the surface. The practical should be repeated so you could work out any anomalies, standard deviation, and also to calculate mean. When sketching the graph, on the x-axis, you will have the sucrose concentration or the solute concentration. The y-axis will be the percentage change in mass, which is calculated by final mass minus initial mass divided by initial mass times 100. You then plot the graph of the percentage change in mass against the sucrose, salt, or solute concentration. There should be a trend showing that as you increase the solute concentration, the percentage change in mass decreases. And then you want to spot where at the x-axis there is 0% change in mass. So in this case, we can see that the solute concentration of the potato is 0.46 moles, because that's where there is no percentage change in mass at that concentration. Therefore, the net movement of water by osmosis into and out of the cells is 0. So by comparing where the line cuts at the x-axis at 0% change in mass for carrot and potato will help us determine the water potential of carrot and potato, so we compare them. State how 1 mole per decimeter cubed sucrose solution can be used to make 0.8 moles per decimeter cubed. Here you'd use an 8:2 ratio of sucrose and water, or simplified, that's a 4:1 ratio. So 4 parts of 1 molar decimeter cubed sucrose solution to 1 part 0.8 moles decimeter cubed distilled water. (4.2) Gas exchange. Explain why single-cell organisms don't have a gas exchange system. Here is a single-cell organism. It's an amoeba, which is unicellular, which means it's only made up of one cell. And in the gas exchange system, they don't have one because they use diffusion to diffuse oxygen into the cell for respiration and carbon dioxide out of the cell as a waste product. As an organism size increases, we can see that the surface area to volume ratio decreases. Therefore, single-cell organisms don't have a gas exchange system because, being a small organism, they have a large surface area to volume ratio, and that means that their surface area for exchanging useful or getting rid of waste is larger in comparison to their small volume of metabolic demands. You can see that the distance from the outside to the center of the cell is very short. So they have a short diffusion distance between the outside and the innermost central area. So that means it's quick to diffuse things from the outside to every part of the cell. And because of the large surface area to volume ratio and the short diffusion distance, diffusion is sufficient enough to supply the needs and get rid of waste for every part of the cell, which means that they don't need a specialized gas exchange surface. In large organisms, because as the size has increased, the surface area to volume ratio has decreased. That means the surface area is too small to exchange useful substances such as oxygen and nutrients and get rid of waste such as carbon dioxide or excess heat in comparison to their metabolic demands of the cells due to their large volume. Also, the diffusion distance between the outside and the innermost cells is too long, so you can't rely on diffusion. It would not be sufficient to supply the needs of the innermost cells and get rid of the waste fast enough. The conclusion from this is, for very large organisms, because of the fact that they have a smaller surface area to volume ratio, they require a specialized gas exchange surface. So, for example, in humans, we have the alveoli, and that increases the surface area to volume ratio. And then once you've increased the surface area to volume ratio in a specialized area, you need to move the molecules quickly to the innermost cells or from the innermost cells. So that requires a mass transport system, such as the circulatory system. So unicellular single-cell organisms or small organisms, they can just rely on diffusion because it's sufficient, but larger organisms, due to the reduced surface area to volume ratio, require the specialized gas exchange surface and a mass transport system to increase the surface area to volume ratio and to move the molecules quickly to the innermost cells. How could you increase the rate of diffusion? You can see on the left image that there is a difference in concentration between outside the cell and inside the cell. So we can see that there is a concentration gradient. But on the right image, you can see that the concentration gradient is much steeper because there is a bigger difference in concentration between the outside and the inside of the cell. Therefore, diffusion should be a lot faster. Increasing the concentration gradient increases the rate of diffusion. You could also shorten the distance, and that will also increase the rate of diffusion. And you could also increase the surface area to volume ratio, and again, that increases the rate of diffusion. Explain how mammals and fish are adapted for gas exchange. Gas exchange happens in the alveoli in mammals, and the idea is to increase the diffusion rate of oxygen into and carbon dioxide out of the capillaries, which contain blood. Using the 3 ideas of how to increase the rate of diffusion that we just talked about, we could use those principles to see how the alveoli is adapted to increase the rate of diffusion in gas exchange. First of all, the alveoli itself has a large surface area to volume ratio due to the folding. The short diffusion distance comes from the fact that the alveoli and capillary walls are thin, or 1 cell thick. And then in terms of the concentration gradient, to maintain the high oxygen concentration gradient between the alveoli and capillary, we have a continuous blood flow. And that means that the blood that is being oxygenated flows away in the capillary such that it takes away the oxygen. So you're always maintaining that concentration gradient between the alveoli and the capillary. Gases also are dissolved inside the alveoli, and this involves a liquid surfactant because liquids dissolve gases. The whole point of the liquid surfactant is that it is moist, and that prevents the collapse of the alveoli. So when you breathe out, the walls generally touch each other, and so when you breathe in again, we want to make sure that these walls do not stick with each other. So to prevent them from collapsing, the surface or the inner surface of the alveoli contain liquid surfactants, and that prevents the collapse of the alveoli. Gas exchange in fish happen in the gills, and again, so you want to use the 3 principles of how to improve or increase the rate of diffusion. So first, to increase the surface area to volume ratio, fish have got gill filaments and lamella in their gills, and that increases the rate of diffusion. They also shorten the diffusion distance by having these gill filaments and lamella that are containing thin walls, and they maintain the diffusion gradient between water and the blood because the water and blood flows in opposite direction, and this is called countercurrent exchange system. In this animation here, if you look at the yellow box, you could see that when you decrease the volume, the pressure increases. So volume and pressure have opposite effects. Decreasing volume increases the pressure, whereas when you increase the volume, the pressure decreases. You could also see on the picture on the right bottom where you've got a muscle that is first relaxed, and then looking at the arrow you could see the muscle that is contracted, and this muscle is wider but more importantly shorter. So when a muscle contracts, it becomes shorter. Outline mechanism of ventilation. Here is a structure of the ventilation system, and ventilation is just a scientific word for breathing in lungs. When we breathe in, this involves, as you can see, the diaphragm moving downwards and the ribs moving upwards. And this requires muscles, particularly the diaphragm, and the external intercostal muscles. So during inhalation, which is breathing in, the diaphragm muscles contract. And as I said earlier, when muscles contract, they become shorter. So the diaphragm therefore moves downwards and flattens. The external intercostal muscles also contract. And when they do that, they cause the rib cage to move upwards and outwards. The total effect of the diaphragm moving down and the rib cage moving up is that the volume of the chest cavity then increases. As mentioned earlier, when a volume increases, the pressure decreases. So therefore the pressure in the chest cavity is now decreased, such that there is a lower pressure in the chest cavity and a higher pressure in the outside atmosphere, and that causes the air to move from the higher atmospheric air pressure to a lower pressure in the chest cavity, so air moves into the lungs. This has the opposite effect when you're breathing out or exhaling. So here the diaphragm muscles relax, and that means that they move upwards and become more dome-shaped. The external intercostal muscles relax, and that causes the rib cage to move downwards and inwards. Therefore, the volume of the chest cavity decreases, and because the volume decreases, that means the pressure will increase. So there will be a higher pressure in the chest cavity in comparison to the outside atmospheric air pressure. So that means air will move out of the lungs from a higher pressure to a lower pressure in the air of the atmosphere. Explain how insects are adapted for gas exchange. Here is a structure of the gas exchange system in insects, and we can see that the air enters through the spiracles, and the oxygen in the air moves from the spiracles to the trachea and then the tracheoles by diffusion. The tracheoles also have a layer of fluid, and that fluid allows the oxygen to dissolve. And then the tracheoles also supply oxygen to the body cells and tissues of the insect. To increase the rate of diffusion, again, we use the three ideas of how to increase the rate. So we've got tracheoles, they provide a larger surface area to volume ratio for diffusion. The tracheoles are also thin, so clearly that's going to shorten the diffusion distance. And then to increase the concentration gradient, the abdominal ventilation movements of the insect move the air that is surrounded, and that maintains a high concentration gradient. Larger insects need to do more respiration, or any insect that is very active also needs to do more respiration. So those organisms are adapted because they have got air sacs that store energy. Explain why gas exchange system in insects limits their maximum body size. Larger insects have an increased distance from air to cells. Therefore, their surface area to volume ratio will be much smaller, and this decreases the rate of oxygen diffusion. Remember, insects diffuse oxygen from the outside through the spiracles, so they don't use a mass flow system to get oxygen to your body cells. They do have blood, but that's mainly to transport nutrients, but not oxygen from the air. Explain how increase in lactic acid concentration will affect gas exchange. Lactic acid from increased activity decreases the water potential of cytoplasm. So when the body cells, specifically muscles, are more active, they produce more lactic acid. And because lactic acid is a solute, that means the water potential of the cytoplasm will decrease. And as I stated earlier, there is a layer of water in the tracheoles, and that means that there will be a higher water concentration on the tracheoles, and that will force that water to move into the body or muscle cell by osmosis. And when it does that, there's less layer of liquid in the tracheoles, and the oxygen should therefore diffuse faster because it's quicker to diffuse through a gas than it is to diffuse when dissolved. Core Practical 7. Dissect an insect to show structure of gas exchange system, taking into account safe and ethical use of organisms. This involved pinning the locus, cutting the body open, and also submerging it underwater to ensure there is clear visibility of the gas exchange structures mentioned above. Describe gas exchange in plants. Gas exchange mainly happens in leaves, and that's because the leaves are thin, and that means there's going to be a short diffusion distance. Leaves are also broad, and that broad means that you're increasing the surface area to volume ratio, and that increases the diffusion of carbon dioxide or oxygen. Within the leaf, there is a spongy mesophyll layer, and these spongy mesophyll layer have got an irregular shape, and that creates air spaces. And that air spaces also increases the surface area to volume ratio for gas exchange. The gases move into and out of the leaf by diffusion through the stomata, and those stomata are surrounded by the guard cells, which open and close. Explain how guard cells change shape during light. First, potassium ions are actively pumped into guard cells when there is a high light intensity. This means that the water potential inside the guard cells is lower, so that causes water to move into the guard cells from the surrounding cell by osmosis. When that happens, the guard cells become turgid, and this causes the thin outer part of the cellulose cell wall in the guard cells to stretch, and they stretch more than their thick inner part, which are less flexible. So when the thin outer part of the cellulose cell wall stretches, that causes the guard cell to change shape, and the somatal pores then open. The opposite effect happens during the night when it is dark and there is a lack of light. The potassium ions are pumped out, the water then moves out of the guard cells by osmosis, but in this case the guard cells now become flaccid. And when they become flaccid, they change shape and the stomatal pore closes. Stomata are not only found on the leaves, they could also be found on green stems, but not on the part that are thickened by woody bark. However, the woody stem tissues and roots can still have gas exchange enabled in them because they have got spaces that develop called lenticels. .3 Circulation. Explain why organisms need a mass transport system. A mass transport system is when you could transport substances in the flow of liquid with a mechanism for moving that liquid. Organisms may also need a specialized gas exchange surface when they increase in size, and that's because the specialized gas exchange surface increases the surface area to volume ratio, because being a larger organism, their surface area to volume ratio is small. Diffusion is inadequate in organisms that have a small surface area to volume ratio, for instance, large organisms. Therefore, a mass transport, such as a circulatory system, can move the molecules quickly between the gas exchange surfaces, such as an alveoli, and cells in comparison to diffusion. The mass transports one directional as opposed to spreading in all directions by diffusion. Explain advantages of double circulatory system over a single circulatory system. In a single circulatory system, such as bony fish, you have blood that circulates from the gill to the body cells and then to the heart. The heart then pumps it to the gills and then again it goes to the body cells and comes back to the heart. So there's just one circulation. Double circulatory system can be found in mammals. Here, oxygenated blood is pumped to the body through the systemic circulation and is pumped at a higher pressure to reach cells quicker. Deoxygenated blood is pumped separately to the lungs in the pulmonary circulation and is pumped at a lower pressure, and that makes sure that the lung blood vessels are not damaged. So the big advantage of having a double circulatory system is that you can have a higher blood pressure than a single circulatory system without damaging the lungs because you pump blood to the lungs at a lower blood pressure. The oxygenated and deoxygenated blood is also split. So oxygenated blood is pumped at higher blood pressure to the body cells and deoxygenated blood is pumped to the lungs at a lower blood pressure. They are pumped separately. And this means that the blood passes through the heart twice. And you can see this in this animation here, where you've got oxygenated blood being pumped to the body cells, and then deoxygenated blood is pumped to the lungs. So therefore you could have a higher blood pressure to the body cells and a lower blood pressure to the lungs, whereas in a single circulatory system, there's just one circulation. So that means that the pressure exerted by the contraction of the heart has to be much smaller so that the gas exchange surface or vessels in the gas exchange surface are not damaged. Describe function of blood. Transport, things like solutes and waste. Also used for defense, for immune responses. And the blood is also used to form tissue fluid and lymph. Describe structure of blood. So you can see here that nearly half the blood is liquid in the form of plasma. It also contains red blood cells, otherwise known as erythrocytes. There are also white blood cells known as leucocytes. We know that white blood cells can release antibodies, but other white blood cells or leukocytes engulf pathogens. So the structure and function of plasma is that it contains water. So that means water we know is a good solvent and acts as a transport medium. They also contain fragments of cells or platelets for blood clotting. Because they contain water, they can transport solutes such as salt, glucose, enzymes, amino acids, proteins such as hormones, lipids, vitamins, mineral ions, and carbon dioxide can be transported using the blood plasma. Red blood cells are known as erythrocytes and they transport oxygen from lungs to body cells. They are adapted to do this because they contain hemoglobin, which binds to oxygen to form oxyhemoglobin. The erythrocytes have no nucleus, and that increases the space for more hemoglobin to be in the erythrocytes. And they also have a biconcave disc shape, which creates a larger surface area to volume ratio, and that means that there is a faster oxygen diffusion into the erythrocytes when they're at the alveoli capillary and out of the erythrocytes when they've reached the body somatic cells. Leukocytes are white blood cells, and there are 4 different types that you've got to learn. You've got neutrophils and monocytes, which can be macrophage as well, and they engulf pathogens by phagocytosis and digest them using lysosomes enzymes. The neutrophils have a multi-lobed nuclei. So if you look at the picture of the neutrophils, you can see that they've got a nucleus that has got more than one lobe. Eosinophils are leukocytes or white blood cells that are non-specific immune response against allergic, inflammation, and parasitic infections such as worms. They have got a 2-lobe nucleus, and that's how you would recognize them if you were to see a picture. Lymphocytes are involved in specific immune response, and they secrete antibodies which bind onto the pathogen's antigen. Examples of these are T and B cells, which you will learn later on. Explain role of platelets and plasma proteins in sequence events leading to blood clotting. Platelets release a clotting factor called thromboplastin. The active enzyme thromboplastin and calcium ions convert prothrombin to its active enzyme form thrombin, and that thrombin catalyzes soluble fibrinogen conversion to an insoluble fibrin. You can see in the diagram that fibrin forms a plug or mesh to seal wound because it traps other cells such as erythrocytes, and that stops bleeding and prevents infection. Explain structure of hemoglobin in relation to its role in transport of respiratory gases. In terms of the structure of hemoglobin, we can see that it's got 4 polypeptide chains, 2 alpha and 2 beta, and each of the polypeptide chains is attached to an iron-heme prosthetic group. The hemoglobin is a globular protein. You can see it's got a 3D shape, which is tertiary structure, and because it's made up of more than one polypeptide, that is the quaternary structure. And because each polypeptide is attached to one iron-heme prosthetic group, that means that it can bind to 4 oxygen molecules, and that is a reversible reaction. So not only can it bind to it, which is associate, the oxygen can then dissociate from the iron-heme prosthetic group. So hemoglobin can bind to 4 oxygen molecules to form oxyhemoglobin. Now how they bind to those 4 oxygen molecules is a bit like the postage stamp analogy of corporate binding. When you got 4 stamps attached to each other, the first stamp is harder to tear off, but then it becomes easier to tear the next stamp. So this is a bit like the binding of the oxygen molecules. The first oxygen molecule that binds to hemoglobin is the most difficult to bind. But then once that oxygen molecule binds, it changes the shape of the hemoglobin protein, and that makes it easier for additional oxygen molecules to bind. This is the same in reverse as well. The first oxygen molecule is the hardest to dissociate and lose. But as the first oxygen molecule is released, it makes it easier to then release the second, the third, and the fourth oxygen molecule. Explain oxygen dissociation curve of adult fetal hemoglobin and myoglobin. Here we've got an oxygen dissociation curve. You can see that it is a sigmoid or S-shaped curve when we look at A for hemoglobin. And when hemoglobin wants to bind to oxygen at lower partial pressure of oxygen, so that means an area that has got low oxygen concentration, such as respiring tissues, we can see that hemoglobin will have a low affinity for oxygen. So in other words, the oxygen doesn't stay binded to hemoglobin, it dissociates from the hemoglobin. And when it dissociates from the hemoglobin at lower partial pressure of oxygen, the hemoglobin are less saturated. The arrow shows that when you increase the partial pressure of oxygen slightly, there is a very large increase in percentage saturation of hemoglobin. At higher partial pressure of oxygen, such as the lungs or gills, because there is a high oxygen concentration, there is a very high affinity of hemoglobin for oxygen. So that means oxygen want to bind to hemoglobin. So therefore, more of the oxygen associates to hemoglobin, and therefore there's going to be a more saturation of hemoglobin. Again, a small decrease in partial pressure of oxygen causes a large decrease in percentage saturation. So as these erythrocytes move to a location that has got less oxygen, therefore the oxygen suddenly wants to dissociate and there will be a large decrease in the percentage saturation of hemoglobin. If you look at line A, which is for hemoglobin, you could shift the oxygen dissociation curve to the right, and when you shift it to the right, you can see that at 2 partial pressure of oxygen, or 2 kilopascals partial pressure of oxygen, originally it was around 43% saturation, but by shifting to the right, it is now 10% percentage saturation. So shifting the oxidation dissociation curve to the right means that there is a lower percentage saturation at the same partial pressure of oxygen. And this happens, or is known as the Bohr effect, because it happens when there is a higher partial pressure of carbon dioxide. So the affinity for oxygen at higher partial pressure of carbon dioxide for hemoglobin is much lower. So in an area that has got a higher carbon dioxide concentration, the oxygen doesn't want to associate itself with hemoglobin. And a good example of that area would be exercising muscles, because they have a higher partial pressure of carbon dioxide. So therefore, when the hemoglobin reaches these exercising muscles, they will lose more oxygen to the muscle tissue during exercise. And that's because you've shifted the oxygen dissociation curve to the right due to the fact that there is a higher partial pressure of carbon dioxide in these exercising muscle tissues. That means the percentage saturation of hemoglobin in oxygen will be much lower, which is good because you want the hemoglobin to dissociate the oxygen so that the oxygen can be used by these respiring exercise muscle tissues. With the oxidation dissociation curve being shifted to the left, this means that there should be a higher affinity, and this happens in fetal hemoglobin. There is a higher affinity for oxygen for fetal hemoglobin. This means that the oxygen transfers from an adult hemoglobin, in other words, the adult hemoglobin in the mother, to the fetal hemoglobin more readily. And that's because the fetal hemoglobin oxygen dissociation curve is shifted towards the left. So oxygen has a higher affinity to fetal hemoglobin than to the adult hemoglobin of the mother. So it wants to dissociate from the adult hemoglobin and associate to the fetal hemoglobin, which is good for the fetus. The same idea happens in myoglobin. So myoglobin is another type of respiratory pigment, just like hemoglobin, but it is found in muscle tissues, and it contains only one heme group, so only one oxygen molecule can bind to it. And you can see that it shifted the oxygen dissociation curve to the left very extremely, to the point where a very high affinity of oxygen to the myoglobin occurs. And that means that it will not dissociate oxygen unless there's a extremely low partial pressure of oxygen. So if that's the only time it will dissociate oxygen, that means the vast majority of time oxygen is associated to myoglobin, and therefore myoglobin acts like a storage of oxygen. It only dissociates oxygen at very, very low partial pressure of oxygen in very active muscle tissue. So this would be a scenario where there's exercise happening, we're running out of oxygen, the oxygen partial pressure decreases and is very, very low, and that's when the myoglobin will dissociate the oxygen to be used. So it acts as an oxygen storage. Here is a picture of 3 different types of blood vessels. You can see they are the arteries, capillaries, and veins. And they've got different structures because of their different functions. The arteries' function is to transport oxygenated blood away from the heart towards the body cells. So you can remember that because artery starts with A, so away from the heart. And when you look at the structure, you could see that it's got a thick wall that's composed of smooth muscle and stretchy elastic fibers, and that helps to withstand the very high blood pressure. Because if blood is going away from the heart, the strong contraction means that there'll be a very high blood pressure, and to prevent the arteries from bursting, it needs to have thick walls, and those walls clearly need to be made of strong smooth muscle and stretchy elastic fibers. The function of the capillaries is that's where exchange of substances with body cells occurs, such as oxygen, glucose, and carbon dioxide. If you want substances to be exchanged, you don't want the blood to flow very quickly because you want to increase the time for exchange to happen. So that's why these capillaries are only one cell thin wall, so they don't have any muscle or elastic tissue. They also have a narrow lumen, just like the arteries. Arteries have narrow lumen because they've got thick wall, but in the capillaries they have got thin wall and a narrow lumen. And the whole idea of having a thin wall and a narrow lumen means that the diffusion distance between the blood and the body cells is short, and that means that there's going to be a faster exchange of substances. The capillaries also need to have walls that are permeable for diffusion to occur. The veins have got a wider lumen and a thinner wall, and that wall has got less muscle or elastic tissue. So because the walls are thin, that means their lumen is a lot more wider. Because the blood flows slowly due to the low blood pressure, there's a possibility that the blood can flow backwards, and that's why veins have got valves. That prevents the backflow of blood due to the low blood pressure in the veins. Describe structure of heart. In mammals, the heart could be split into 4 different chambers. The top chambers are called atriums, and the bottom chambers are called ventricles. Oxygenated blood flows in the left side of the heart, whereas deoxygenated blood flows on the right side. When blood goes from one chamber to the next, it has to go through valves. So from the left atrium to the left ventricle, there is an AV valve, and the same idea from the right atrium to the right ventricle. To leave the heart, there are also valves. So the left side of the heart is where there is flow of oxygenated blood, and that enters the heart through the left pulmonary vein. As it's a vein, that means blood is going in to the heart, and because it came from the lungs, it is the pulmonary, and because it's the left side of the heart, it is oxygenated blood. So oxygenated blood flows into the heart through the left pulmonary vein into the left atrium. Top chambers are called atrium, this is the left side, and blood always enters the atrium. Then the oxygenated blood flows from the left atrium to the left ventricle, and it has to go through an AV valve, and this AV valve is called the bicuspid valve. And then when it leaves the left ventricle, again it has to go through a valve, and this is the semilunar valve. And then when it goes away from the heart, that will be an artery, and this artery is the biggest artery in the body, known as the aorta. On the right side of the heart, we've got the flow of deoxygenated blood. Again, blood enters the heart on the top chamber, so this will be known as the right atrium. And because it's going into the heart, it's a vein, and this is the biggest vein, known as the vena cava. And that deoxygenated blood then flows from the right atrium to the right ventricle through another AV valve, and this AV valve is known as the tricuspid valve. The blood then flows out of the right ventricle through a semilunar valve, and then it is contracted and pumped towards the lungs, and as it's going away from the heart, it's called an artery, and because it's associated with the lungs, this is the pulmonary artery. All the valves, whether it's the semilunar valves where the blood leaves the heart, or the AV valves, the bicuspid, the tricuspid, these valves, just like in veins, prevent the backflow of blood. The heart also has a septum, which is like a wall in the middle, and the whole point of the septum is to prevent oxygenated and deoxygenated blood from mixing. Also notice that the left ventricle muscle wall is a lot more thicker than the right side. And because it's more thicker, that means it contains more muscles. And if it contains more muscles, that means the contraction is going to be much stronger. And because it's a stronger contraction, that means you could pump the blood around the whole body a longer distance and much faster. So that means that the contraction and the pumping of the blood will be under a higher blood pressure, and that helps the blood reach cells further away much quicker. Right ventricle muscle wall is much thinner, and that's because the blood only needs to be pumped to the lungs, so it doesn't need a strong contraction, otherwise the blood vessels in the lungs will be damaged. Describe sequence of events for cardiac cycle. Here is a diagram of the cardiac cycle, and you need to know the structures of the heart to understand how the cardiac cycle works. And first of all, the heart itself doesn't require an external nerve impulse to contract. It could contract on its own. So it does not require an electrical impulse to be received from the brain to contract. So when it stimulates its own contraction, this is known as myogenic. So the heart is myogenic, it can stimulate its own contraction within the heart. This happens in the heart's pacemaker cells. And that pacemaker cells, they are located in the right atrium in a location known as the SAN. So that's where the contraction is initiated by the heart on its own. That electrical impulse from the SAN, due to the contraction, is sent to the rest of the atria, both the left and right, and that causes the atria to now contract. So we say that depolarization from the SAN causes the atria to contract. That contraction is then delayed at the AVN, and the reason for delaying it is because as the atria contract, both the left and the right, we don't want the ventricle to contract at the same time. So having that delay means that only the atria will contract first and the ventricle could contract later on. Therefore, the blood doesn't go directly from the atria to the ventricle and out of the heart in one go, which will mean that the blood pressure will be too low. We want the ventricles to contract to pump blood out of the heart. So the atria should only contract to the ventricles. So the delay at the AVN means that the ventricle contraction happens afterwards. The depolarization then goes through the bundle of His until it reaches the apex of the heart, and then depolarization causes the ventricles to contract, and this is known as ventricular systole. And that is the sequence of events for the cardiac cycle for one heartbeat. Explain stages that lead to atherosclerosis, which is the hardening of arteries, its effect on health, and what are the factors that increase the risk of its development. First of all, the endothelium becomes damaged, and this could be due to modifiable lifestyle risk factors such as having a high blood pressure, smoking, stress, or raised LDLs. This causes an inflammatory response, and white blood cells or macrophages then arrive at the location, and they cause deposits of cholesterol or calciums to build up in the walls of the artery. This forms atheroma or plaque or atherosclerosis, which then blocks the arteries. The arteries narrow, which reduces the blood flow or the oxygen supply or the glucose supply to the cells. Because of this, that means the heart contracts much stronger, and that increases the blood pressure. And that could lead to an heart attack because the heart muscles itself doesn't receive enough oxygen or glucose. It could also lead to stroke. In this case, it's the brain cells not receiving oxygen or glucose. It could also lead to angina, thrombosis, blood clots, or aneurysms. Non-modifiable risk factors for atherosclerosis include genes, age, and sex. Interpret data showing ECG traces and pressure changes. If you look at the ECG trace at the bottom, ECG is an electrocardiogram, and it's related to the contraction of the heart. There are 3 stages that you need to know for the exam. You've got P waves, QRS complex, and T waves. And going back to the cardiac cycle, we could see that we could split that into those 3 distinctive stages. So a P wave will show the depolarization of the atria, where you've got atrial systole and the atria then contracts. The QRS complex of the ECG trace shows depolarization of the ventricles during ventricular systole. So that's when the ventricles will contract. And the T wave of the ECG trace is when there is repolarization of the ventricles. That's when the muscles are relaxed. When interpreting these ECG traces, you could see on the top corner right ECG trace, you've got the normal heartbeat, and then below that, you've got a fast heartbeat. And in the fast heartbeat, you could see that the intervals between the waves are much shorter, whereas in a slow heartbeat, those intervals for the waves are much longer. And then in irregular heartbeat, you can see that there are different length intervals for the different waves shown on the ECG trace. And that's how you could identify and interpret them. And the different parts of these waves can then be linked to the pressure changes that happen in the heart. So we know that when the volume increases, the pressure decreases, and when the volume decreases, the pressure increases. Explain why semilunar valves close. This is because when the blood leaves the semilunar valve on the left side of the heart, it flows into the aorta. The pressure in the aorta, because blood is flowing towards it, is higher than in the left ventricle that it's just left. During the ventricular diastole, the ventricles obviously are now relaxing, and that's forcing blood around the body rather than the blood flowing back into the ventricles. So because there's a higher pressure now in the aorta, that means that causes the semilunar valves to close. So the blood doesn't flow backwards. And you can see these phases of the cardiac cycle. So at the beginning, you've got the P wave, you've got atrial systole, where the atria contracts and forces blood into the ventricles. Then you've got the QRS complex, which is the ventricular systole, and that's when the ventricles contract, and that pushes the AV valves to close due to pressure changes. The semilunar valves then open and blood goes out of the heart, whether it's the left or the right side. Then you got ventricular dystole during the T wave, and that's when there's repolarization, so those muscles are now relaxed for the next heartbeat to occur. And this animation shows the changes in the cardiac cycle, linking that to the ECG trace. You can also see that on this animation, which also includes the changes in the pressure. When we're talking about blood vessels, we've got oxygenated blood in the artery, and that oxygenated blood then flows into the capillary where there are body somatic cells. The oxygen diffuses to these cells for respiration and carbon dioxide diffuses out, and we said the blood is now deoxygenated, and that then moves or gets transported into the vein. So when you look at the capillaries, we've got an artery side and we've got a vein side. Explain how interchange of substance occurs through formation and reabsorption of tissue fluid. So this diagram will help us understand how tissue fluid is formed and reabsorbed. So here you can see this is a capillary, and we've got on the left side the artery side of the capillary, and then on the right side of the diagram this is the vein side of the capillary. And this capillary is surrounded by the body somatic cells. There is hydrostatic pressure that comes from the veins into the capillaries and then into the arteries, then the capillaries, then the veins. And this is because the hydrostatic pressure comes from the contraction of the heart that pushes the blood to move from the artery to the capillary and then the vein. You could see that the highest hydrostatic pressure in the capillary is at the beginning when it's closer towards the artery, and then that hydrostatic pressure decreases as it moves closer and closer towards the vein. There's also an oncotic pressure in the capillaries, and that's caused by the fact that the capillaries contain plasma proteins. And because of these plasma proteins, which can dissolve in the liquid of the blood, that means the water potential of the capillary is lower, and that means there's a higher water potential sometimes in the tissue fluid surrounding the body cells. So that means water wants to move from a higher water potential in the tissue fluid surrounding the body cells towards the lower water potential in the capillary. And that creates an oncotic pressure or an osmotic pressure that wants to force the fluid out of the tissue fluid and into the capillary. So you've got two types of pressure acting against each other. And the oncotic pressure is stable or is at the same rate throughout the whole capillary. Now the capillary side that is closer to the artery, you can see that the hydrostatic pressure is much higher than the oncotic pressure. So if the pressure due to the heart contraction is stronger than the pressure from osmosis, that means that more of the fluid, which contains water and low molecular solutes, is going to be forced out of the capillaries and it's going to move into the tissue fluid surrounding the body cells. And that forms the tissue fluid. On the side of the capillary that is close to the veins, you can see that the hydrostatic pressure is lower than the oncotic pressure. And that means the tissue fluid that is surrounding the body cells would therefore get reabsorbed back into the capillaries. And this tissue fluid usually contains less oxygen and more carbon dioxide because the oxygen has diffused into the body cells and carbon dioxide is diffused out after respiration. So therefore, moving back into the capillary, it will contain more carbon dioxide. Describe what happens to tissue fluid that isn't reabsorbed directly to blood. Approximately 90% of the tissue fluid gets reabsorbed back into the capillaries. However, those 10% that isn't reabsorbed directly to blood, excess tissue fluid is absorbed by the lymphatic system, which then drains into the veins later on. Explain why production of tissue fluid is vital for cells in the human body. That's because when you produce these tissue fluids that comes from the capillaries, it supplies the body cells that it bathes and surrounds oxygen and glucose for respiration, or amino acids to synthesize proteins, fatty acids, salts and hormones. All of these stuff help the cells to grow. The tissue fluid, as I stated earlier, also helps to remove carbon dioxide or remove urea, also prevents the cells from being poisoned. Compare and contrast transport of fluid in vein with its transport in lymph vessel. Similarities: both the vein and the lymph vessel have a low pressure. They also have valves to prevent the backflow of the liquid, and they use muscles surrounding the vein or lymph vessel that squeeze the fluid forward. Differences: in the vein, the flow of the blood is much more faster. That's because of the contraction from the heart, causes the hydrostatic pressure. That flows through the heart in the vein, but in the lymph glands, the excess tissue fluid flows towards the lymph, which then drains into the veins in the future. And here you can see the artery side of the capillary and the vein side of the capillary. You can see that you've got a higher hydrostatic pressure from the arteries towards the vein. So on that side, tissue fluid is produced. So the fluid from the capillary goes into the tissue fluid that surrounds and bathes the body somatic cells, whereas closer towards the vein, the oncotic pressure, which is the pressure from osmosis, is higher than the hydrostatic pressure. Therefore, that tissue fluid gets reabsorbed back into the capillary. 4.4 Transport in plants. This is an experiment you might have done previously using celery and dye, and you can see that the dye goes up the celery through the specialized cells called xylem cells. And the xylem cell is part of the vascular bundle that also includes phloem. They're both involved in transport, and the xylem transports the colored dye, which contains liquid water. So therefore xylem is involved in transporting water. And if you look at the xylem cells on the picture at the bottom right, you can see that these cells are much more wider in comparison to the phloem, but they are located very close to each other within the vascular bundle. Explain structure of xylem and phloem tissues in relation to their role in transport. So here is the xylem, and there's one xylem cell, and you can see that these xylem cells are wide, and that transports large volumes of water, but they also contain minerals which are dissolved in the water. To transport as much water as possible, these xylem cells have got no cytoplasm. So in other words, they're hollow. And because they have no cytoplasm, which means that there's no reactions happening within the cells, that means they're going to be dead cells. So they're hollow dead cells with no cytoplasm. Also, between one xylem and another xylem cell, they have no end walls. So there's no cell wall between xylem cells, but only between the xylem cells. There are cell walls on the outside. And the whole point of having no end walls between the xylem cells and making them hollow means that more water can clearly flow through. That means you're reducing the resistance of the flow of water. So there is a low resistance to water flow due to the fact that they're hollow and there's no end walls between the xylem cells. As the water flows through, the water collides with the cell wall on the side. And when it collides with it, it causes pressure from the water. And that could cause the xylem cell to burst, which is known as lysing. So that means that there needs to be cell walls there, and these cell walls are made of cellulose, and they're much more thicker, which strengthens the cell, but they also have the addition of lignin, which further strengthens the cell. And that strengthens the wall to stop the xylem wall or xylem cell wall from collapsing. Water from the xylem could also flow sidewards, and this occurs because they are perforated by pits. The xylem transports water and minerals, but there are also phloem close by that transport sucrose. In the phloem, you've got sieve tube elements with sieve plates between the cells, and they've also got enlarged plasmodesmata. So having those sieve plates between the phloem cells and having plasmodesmata means that contents can flow through from one cell to the next. Also, the cytoplasm has very few organelles, and that reduces the amount of resistance for the sucrose, which is dissolved, to flow through. Because there are fewer organelles in the cytoplasm, that means that there can't be a lot of possibility for chemical reactions to occur. So that means that these sieve tube elements cells need support, and they get support metabolically from the phloem companion cells that are nearby. Explain how water can be moved through plant cells. In other words, what is the pathway taken by water after they enter the root hair cells or before they enter the xylem? And there are two different pathways. There's the symplast pathway and there is the apoplast pathway from the root hair cells to the xylem. And we compared them. The apopass pathway uses the cell walls and the intercellular spaces for the water and the minerals to move through. This is a non-living method. It's faster because it's less resistant, and it's a passive method using diffusion. However, to reach the xylem, it gets blocked by the Casparian strip. In the simplest method, the water and minerals flow through the cell membrane, the cytoplasm, and the plasmodesmata. So this is through a living pathway, which is slower due to more resistance, and the water moves by osmosis in cytoplasmic streaming. It doesn't have to cross the Casparian strip. You can see both pathways here. You've got the apopass pathway, which moves through the cell walls and through spaces between cells, less resistance through a non-living pathway, whereas the simplest pathway moves through the cell membrane, the cytoplasm, and the plasmodesmata. That means there's going to be more resistance. However, this is through a living pathway, and it doesn't have to be blocked by the Casparian strip. Here's an animation showing transpiration of water moving from the roots through the xylem all the way to the leaves and then being evaporated out of the stomata. What is transpiration? In transpiration, you've got the movement of water from the roots to the leaves. You can see the water then goes out the stomata, and when it goes out the stomata, it goes out as water vapor. So to convert from water to water vapor, it has to evaporate, but then to move out the stomata, it has to diffuse out. So it evaporates and diffuses out the stomata as water vapor. Explain how cohesion tension model explains the transport of water from the plant roots to the shoots. So first, you could see that the water evaporates and diffuses out of the stomata. This creates a water potential gradient because when water evaporates out of the stomata, there's a low water potential in the area where the water evaporated out, and the surrounding cells have a higher water potential. So this causes water to move from a higher water potential on the surrounding cells to a lower water potential in the cell that has just lost water that's been evaporated. So those cells will now have lower water potential due to the evaporation. So they need their water to be replaced. And that water is replaced through the transpiration stream in the xylem. The water is being pulled up, and the water gets pulled up in the xylem in the transpiration stream because these water molecules form hydrogen bonds with each other because of the fact that they're polar. Because they're polar water molecules, they form cohesion between these polar water molecules due to the hydrogen bonds. The water molecules could also form hydrogen bonds with the xylem walls, and this is called adhesion. Explain how light, temperature, humidity, air movement affects rate of transpiration. If we do that, there's a high tensile strength in the column of the water in the transpiration stream, and because there's a high tensile strength due to the hydrogen bonds between the water molecules, that's why they called it the cohesion tension model. It's cohesion because cohesion between the polar water molecules, and tension model because there's a high tensile strength within the transpiration stream of the xylem. Okay, how do factors affect the rate of transpiration? When light intensity increases, the rate of transpiration will also increase. And that's because when there's more light, plants want to do more photosynthesis. And when they want to do more photosynthesis, that means they need more carbon dioxide. So that means they're going to have to open the stomata. And when they open the stomata, more carbon dioxide can diffuse in, but that means that more water can diffuse out. So when there's a higher light intensity, the rate of transpiration increases because more stomata will open. And that means that there's going to be an increase in evaporation and diffusion of water vapor out the stomata. In terms of temperature, when you increase the temperature, the rate of transpiration increases. That's because when you increase the temperature, the water molecules will have more kinetic energy, and because they have more kinetic energy, they're going to move much faster. And if they move much faster, that means there's going to be an increase of evaporation, diffusion of water vapor out of the stomata. In terms of humidity, when does humidity increase the rate of transpiration? That's when humidity decreases. So when you decrease humidity, the rate of transpiration increases. That's because an environment that is more humid has more water vapor outside the leaf. So if there's more water vapor outside the leaf, the difference between inside the leaf and outside the leaf is less. That means the concentration gradient will be lower. So having a lower humidity means that the concentration gradient will be increased. And that means that there'll be more evaporation, diffusion of water vapor out of the stomata. When air movement increases, the rate of transpiration increases, and that's because the air movement or wind will blow the water vapor away from the leaf. And when it blows the water vapor away from the leaf, that means there's a bigger difference in concentration within the stomata and outside the stomata. So that concentration difference means that the concentration gradient increases, and that increases the evaporation, diffusion of water vapor out of the stomata. So here's an example of how concentration gradient work. You can see two different concentration gradients. Here you've got a high concentration and a lower concentration, and therefore these particles are going to diffuse from a higher to a lower concentration down a concentration gradient. But on the picture at the bottom, you can see that there's a bigger difference in concentration between the higher and the lower, and therefore the rate of transpiration, the rate of diffusion will be faster because there is a steeper concentration gradient. So these particles will diffuse down a concentration gradient much faster. Using those ideas, we could discuss wind and humidity again. We can see that increasing the concentration gradient increases the rate of diffusion. So when there's air movement that's increased, or when it's more windy, the wind will clearly blow the water vapor away from the leaf, and when it blows it away from the leaf, that means there's going to be a bigger difference inside the leaf and outside the stomata. So you're increasing the concentration gradient, and that means the rate of transpiration increases. In terms of more humidity, when it's more humid, there are lots of water vapor outside the leaf, so that means the concentration gradient decreases. So decreasing the humidity, so when there's less of a water vapor outside the stomata, that means the transpiration rate will increase because you're increasing the concentration gradient, and therefore there's going to be more evaporation and diffusion of water vapor out of the stomata. In core practical 8, you've got to investigate factors affecting water uptake by plant shoots, and this can be done to investigate transpiration using a bubble pottometer. To describe how a bubble pottometer can be used to investigate factors, first you've got to change factors. So you could use things like a fan to change air movement, or a lamp for light, or heater for temperature, or you could actually put a bag around the leaf and that increases humidity. You go have a method that describes how you'll change an independent variable for at least 5 different values. And then you also want to state the control variables. So whichever one you're changing, all the others are the control variables. So if you are changing the light intensity, you'd keep the temperature and the humidity the same. And also the air movement. And then in your bubble potometer, there'll be a bubble, and as the water goes into the leafy shoot, it will then transpire out of the stomata, and that causes the bubble to move to replace the water that is moved into the leafy shoot. You can then measure the distance the bubble moves. You also need to measure the time taken, and that's because you could then calculate a rate. So the rate should be the distance moved divided by the time taken. So that could be in centimeters per minute. But you could also convert that distance into the volume, which I'll discuss later on, which will be more accurate, and that's centimeter cubed per minute. You also want to take some repeat readings, and that helps you to calculate a mean. And you could do repeat readings in a bubble potometer by using the syringe or the reservoir or the three-way trap, which pushes water back into the bubble potometer, and that resets the bubble back to the scale, so you could redo the practical again. Explain how to convert readings measuring distance that air bubble moved into transpiration rate with units millimeter cubed per centimeter squared per minute. So the millimeter cubed is a volume, and that must be the volume of the water going into the leafy shoot, and that could be calculated because you know the distance the bubble moved. So by looking at the distance the bubble moved, you could then multiply that by the area of the circle of the potometer. So that would be pi r squared multiplied by the distance the bubble moved, and that will give you millimeter cubed, as long as you measure it in millimeters in terms of the distance the bubble moves. To do it per centimeter squared, that will be the area of the leaves. So clearly if you have more leaves, there'll be more transpiration. So millimeter cubed per centimeter squared, that the centimeter squared is the total area of the leaf, and then the per minute is by dividing it by time. Explain how to produce accurate readings when investigating effects of factors on transpiration. So how could you make this experiment more accurate? When you cut the stem of the plant, you could do that underwater, and that means that you're reducing the chance of air bubbles blocking the xylem, and that maintains the transpiration stream. The other thing you could do to prevent water loss from the apparatus, which is a similar idea, is obviously to seal the rubber connection. And that rubber connection means that the water molecules are not being lost, and that maintains the cohesion between the water molecules as it's moving through past the rubber tubings and into the leafy shoot. Explain mass flow hypothesis movement of sugars through phloem tissue. First, you need to have a source of sugar. So clearly the source of sugar is the green leaves and stems. Then that sugar needs to be transported somewhere. So it's usually transported as sucrose, and we say that the area that needs the sucrose is called the sink, and that could be a dividing meristem area such as the tip of the roots or the tip of the shoot, but also other parts of the plant that might want the sucrose are things like fruits, seeds, or storage organs such as potatoes, because they do not do photosynthesis. In the source, there is a high sucrose concentration and a low water potential. And in the sink, you've got a low sucrose concentration and a high water potential. At the beginning, you want to move the sucrose from the source towards the phloem sieve tube elements, and that's called phloem loading. So phloem loading the sucrose from the source to the phloem sieve elements, and that occurs through two different pathways, which we learned earlier. We learned in transpiration, there's two different pathways for the water and minerals to move, called the symplast and apoplast. So this is the same idea. In translocation, you can move sucrose through the symplast and the apoplast. So you've got the symplast, which is through the cell membrane, cytoplasm, and the plasmodesmata, or the apoplast, which is through the cell walls and the spaces between the cells. Sometimes to move the sucrose to the phloem sieve elements requires the use of ATP to be which provides accessible source of energy produced from respiration. And this means that you're transporting the sucrose by active transport from the companion cell to the phloem sieve tube elements. This creates a really high sucrose concentration now in the sieve tube elements where there is lots and lots of sucrose. And because it's got a high sucrose concentration, that means the water potential of that area is really low. And this causes water to move from the xylem into the phloem sieve elements by osmosis. And when the water moves from the xylem to the phloem sieve tube elements, from a higher water potential to a lower water potential in the sieve tube elements, that produces a hydrostatic pressure that moves the sap by mass flow towards the sucrose sink. And when it moves towards the sucrose sink, the sucrose can then be moved out of the sieve tubes and into the sink, and this causes the water potential of the sap now to increase. And when it increases, that means the water can now move out by osmosis back to the xylem down a water concentration gradient. Explain why phloem is involved in transport of sucrose. The reason why we involve the phloem in plants is because the phloem can transport molecules down the plant, taking sucrose from the leaves to the roots. That's because the sucrose can dissolve in the water, and you can use mass flow to transport that sucrose. It's a lot more faster than using diffusion.